All right, we are going to go ahead and get going. So it is my pleasure to introduce someone very special today. This is Dr. Meg Daly. She is a professor in the Department of Evolution, Ecology, and Organismal Biology at The Ohio State University. Don't forget the V. And Meg was my PhD advisor. And so this is her first trip down to Dauphin Island Sea Lab. And Meg's career has spanned well over 20 years of working on all aspects of sea anemone evolution, systematics, and diversity. And she is my scientific role model and the best scientist that I know. So please welcome Meg Daly to Dauphin Island. Thanks.
Thank you all. I'm just going to take a second to adjust everything here and make sure I know how the tech works. Okay. Is everything okay with this on the Zoom side too? Okay. Fantastic. So, as I said, thank you. Thank you so much for being here. Today, I'm going to talk about one aspect of sea anemone biology. When I was thinking about what I wanted to talk about, knowing that you all know about the fantastic work that Ben and his team is doing was kind of a challenge. I usually go places and people don't know anything about anemones, and I can talk about kind of anything I want, and then in the realm of anemone biology. But here I'm able to talk about a kind of narrower and more specific thing because you all know a lot about anemones from having you're really one of the main focal points of sea anemone research in the world. And so it was a real pleasure to think about what I was going to talk about, and I'm going to talk about venom because even if you don't know sea anemones, let's see, advancing this. Okay, let's see. I knew there'd be something. Okay, there we go. There we go. Even if you don't know very much about anemones, you know that the world is a dangerous place. You're all out here in nature in this beautiful, much more beautiful than Central Ohio environment. And so as people who hike or swim or fish or do field work, you know that there are many, many, many different ways in which nature is out to get you. Shakespeare claimed nature was red in tooth and claw, but it is also full of fangs and stingers and spines. The world is a dangerous place. It's so interesting that what I'm seeing here and what you're seeing there are totally different. So when we think about venomous organisms, I always start by trying to level set. Poisonous are things you bite it and you get sick. Venomous things, they bite you and you get sick. Venom must be injected into the target. Venom is a complex series of biomolecules. Most organisms have what we call a venom cocktail, and that venom cocktail includes many different kinds of compounds that may have many different effects. So we could have everything from cytotoxins, which punch holes in cells and just generally destroy cells. You could have toxins that dysregulate the blood system, often through changes to clotting. You could have toxins that cause immune responses. You could have toxins that cause metabolic dysregulation, insulin-like toxins. And these are useful, for example, making animals sleep and be more immobilized as you're trying to consume them or if you're trying to evade them. And most commonly studied are neuromuscular toxins that are paralytic or otherwise impacting the neuromuscular system. Neuromuscular toxins, even when we think about a broad class like neuromuscular toxins that are all acting in largely the same way, they can impact this. This just shows an example of all the different ways that a neuromuscular toxin might act. It could be acting on the neuron, it could be acting on the junction, it could be acting on the muscle. Different kinds of molecules target different parts of this complex pathway. So venom is well studied because it is ubiquitous among animals. What makes venom such a compelling trait is that everybody does it. So this is a phylogeny of animals, and where the end lineages are red or blue, those taxa make venom. So this is everything from cnidarians to spiders to worms to snails to starfish and sea urchins to snakes to fish and includes things that you might not think about as venomous. For example, these lorises are actually venomous. They're venomous mammals. They use modified glands that produce toxin. It drips down a groove in their arm and it's injected with a special claw. So there are venomous mammals. And this one looks so cute, right? Like I would totally want to snuggle that little mammal, but it has means of protecting itself from people like me who want to snuggle it or other animals that want to eat them. So venom is this animal-wide trait. It's a common strategy. And so for me as an evolutionary biologist, why I got interested in venom is because I'm interested in things that happen lots of times in evolution. How and why do animals keep hitting on this same strategy? And many evolutionary biologists are drawn to these questions of repeated evolution and when the circumstances under which things evolve repeatedly, the subtle differences when traits evolve differently, and there are kind of two large categories of explanation for convergent repeated evolution. And one is deep ancestry. And the best studied example here are eyes. So eyes occur multiple times across animals. It seems that at least some elements of eye evolution kind of pop up and go away. So it's not like the amniotic egg, which evolved once and then was retained by all the descendants. It's a trait that shows a lot more dynamism in evolutionary history in terms of the way it is expressed and whether it is expressed. But eyes, we think, are an example of deep ancestry. So even though there are differences among fish eyes and human eyes and frog eyes and fly eyes, there are also commonalities. And that commonality comes through shared sets of genes that change in their regulation, in their makeup, in terms of the sequence composition, in terms of the copies. And that shared genetic pathway underlies this common expression of eyes. And so the different manifestations are just turning on, turning off, or modifying this pipeline of information that is deeply shared among animals. Another kind of repeated evolution is what we see in things like wings. There is no evolutionary relationship between the wings of dragonflies and the wings of birds or bats. They come from completely different embryonic tissues. Actually, everything that's happening in insects is upside down relative to everything that's happening in vertebrates. So they are completely unrelated in terms of their evolutionary origin, whether they're made of muscle and bone or cuticle. All of it is different. Even in cases of birds and bats, which are pretty closely related at a broad scale, although they are both forelimb modifications, it's pretty different in terms of the elements of the forelimb that is being modified. So we have these two different modes of thinking about convergence. And so for me, when I started studying venom about 10 or 15 years ago now, this was really the question that I was interested in. Is venom deeply homologous or convergent? And then specifically, what's going on in cnidarians? So venom, as I said, requires an injection mechanism. And broadly speaking, there are two kinds. There are fangs and spines. So fangs are usually associated with the mouth. They're usually associated with some kind of salivary gland or oral secretion that is part of injection. And we see fangs in snakes. We see fangs in things like leeches. We see fangs, oral harpoons in a lot of mollusks. Or we could think about spines, often more distributed across the body, many smaller individual glands, things like fish. They're generally spines that are injecting venom. There are spines in sea urchins that are part of venom, and in things like fireworms, which as marine scientists, many of you have probably encountered the venom of fireworms. And what's interesting about this is that the lineages having spines and the lineages having fangs, it's all over the place, right? There's two worms up there. Some worms have spines, some worms have fangs. So there's not a necessary correspondence, even when there is venom in a single lineage, in the delivery mechanism. So that's an opportunity for evolution to be acting and shaping venom phenotype. The other way that evolution can shape the venom phenotype is through all of the different compounds that are part of it, all of these biomolecules. So this is just showing broad categories of these neuromuscular toxins. This is that diagram I showed you before. And you can see within lineages, like everything that a cone snail makes is flagged with a tiny little cone snail. Things that are made by spiders are flagged with a tiny spider, urchins and jellyfish up there. You can see cone snails make multiple kinds of sodium channel blockers. They also make nicotinic receptor antigens or antagonists rather. They're making they're monkeying with all different parts of this pathway. It's not just a one-to-one relationship. They are making compounds that interfere with this whole pathway. And as an evolutionary biologist, this is fascinating, right? Why so much? Why are they such haters, right? Why not just make one kind? Why are they making so many kinds, so many different kinds of neurotoxins? And this is just one. They're also making cytotoxins. They're also making insulin dysregulation molecules. They are going in many ways, right? They're going overboard. They're just really having at it. So there's all these opportunities for variation, for evolution to be acting independently in different pieces of these systems. Generally, what we see is two different styles of approach where organisms are repurposing genes in gene families that are related to the function, right? So they are repurposing neurotransmitters to dysregulate the system, like going rogue within the system that they evolved to function in. Or they're weaponizing genes that function in other pathways. So a lot of the cytolytic toxins are related to genes associated with programmed cell death and kind of cleaning up cell function, so apoptosis. So you can either take things that do that function, but in a good way and weaponize them, or you can take things that are participating in a system and help them go rogue. And as I said, it's ubiquitous among metazoans, which is a really exciting evolutionary story. But what's really, really cool when you look at this tree is what's happening here at the base. So that lineage, jellyfish, sea anemones, phylum Cnidaria, and it's colored red. All of the other lineages, you can see there's things that are red, there's things that are blue, there's things that are black. So venomous, not venomous, different kinds of venom. All of those lineages, are equivalent evolutionarily to Cnidaria. Cnidaria is the sister group to triploblasts. Triploblasts are shown there in many different exemplars within that lineage because they do all different kinds of things. Cnidaria evolved so that the entire lineage is venomous. It is the only phylum level lineage among animals where every single member is venomous. It is also the oldest lineage in which venom evolved. So there is an opportunity to understand venom within Cnidaria as a way of kind of snapshotting all of what might be happening in its sister group. And also, there's something weird going on. There's something different about venom, or there could be something different about venom in Cnidaria because of these aspects of it. So what I'm going to talk to you about today is what is happening in terms of venom evolution in Cnidaria, really focusing on sea anemones. And the structure of the animal tree of life really provides us with this opportunity. So we've got venom originates one time, is never lost. Venom originates 5 to 15 times and is lost a lot. So really a different paradigm. So if we take that as our starting place, what differences then do we see? More, in terms of structures and in terms of the biomolecules. So I just want to highlight a couple things about venom. This is Cubomedusin, a box jellyfish, highly toxic but tiny. That's the head of a match, and you can see that the head of the jellyfish there. So the venom of the Chironox, of the box jellyfish, is among the most deadly of all the organisms. It's about as deadly as the reef snake, but the animals are tiny. So the reason that jellyfish, that cnidarians aren't killing people left and right is because although they make super potent venom, they don't make a lot of it because they're little by and large relative to the size of people. And so if there were a swarm of jellyfish, or if you were a small person, like a child, or if you were physiologically challenged in some way, this is likely to be very harmful to you. If you are a healthy adult, it is unlikely to be life-threatening to you. So one of the things about cnidarian venom is the animals make small quantities. They also make their venom in a distributed system. So unlike snakes, which have fangs and a gland behind it, or cone snails, which have a harpoon and a gland behind it, or scorpions, which have a spine and a gland behind it, cnidarians have venom all over their body in small amounts in secretory cells and encapsulated within their nematocysts. The nematocysts, which are the most complex thing a cell makes. I always imagine that cells dream of making nematocysts, and only some cells have that dream realized because they are hyper complex machines. It's like Golgi bodies. This is what they do when they get really, really lucky and they're born into a cnidarian. They get to make these complex micro machines that are, tiny, microns large, but that are filled with venom, and that also when they are discharged and punch through the body of the target, create a pathway through which secreted toxins can also enter. So just like the fang creates a hole and then the venom in the venom gland enters into the target, the nematocyst makes a hole and secreted venom, not just that within the nematocyst, but venom made in cells adjacent to it, can enter into the target. And this doesn't happen in one place. It happens all over the body of the animal. So this is just a pretty old school picture of a hydrozoan polyp. And you can see there are nematocysts all over it, and just some pictures of what nematocysts look like. These are scanning electron micrographs. And you can see the capsule and the thread. And when the nematocyst is not fired, the thread is highly, highly, highly coiled inside the capsule. The capsule has fluid in it. We think that fluid is venomous. And when it's discharged, the thread pops out, pokes into the body of the target, and a little bit of venom comes out with it. But then other compounds enter through that hole. So here is what capsules look like when they're not fired. And here's just some examples of variation. So a ton of variation in these structures. So nematocysts are the quintessential attribute of cnidarians. They evolved at once. No one has lost them, but they've been modified a lot in terms of the structure and function. In fact, some seem not to be piercing. So the one on the bottom there that has the little coils on it, those seem to be modified so that they grab onto the hairs of microcrustaceans and other things. You can see a hair next to it. They're like lassos. So are those even venomous anymore? We have no idea. They seem not to function in the way we would expect in terms of piercing the body, the integument of another animal. So a lot of variation. Other ones are like clearly that harpoon one. Your own experience tells you that that is probably, punching through a pretty thick integument. So this diversity in nematocysts actually raises a question about the homology of structure for delivery of venom. And this is actually the homology of nematocysts is a very long-standing question in the 19th century as microscopy kind of took off. Lots of biologists asked these questions, but they were essentially unanswerable, right? You can observe nematocysts in cnidarians. You don't observe them anywhere else. The inference is that they're homologous. But in trying to connect all of these morphologies, we're kind of lost. No one has made much progress on that. And so we assume they're homologous because we can map a single evolutionary origin of them, and they're made of similar materials. We don't know if they're actually assembled the same way. We have good data on the assembly of nematocysts in Hydra, and that's it. So whether or not all the other kinds assemble the way that Hydras do, we don't know. There's a lot of structural diversity. There's diversity in discharge. So it's possible that there is more complexity here than we interpret. Interestingly, the advent of sophisticated molecular biology tools has allowed us to ask this question. And like all cool science, it happened by accident. This is work led by Leslie Babonis at Cornell, and Leslie was making some fancy nematostella, which is our lab rat sea anemone, using CRISPR. And what she noticed when she looked at these animals is they had weird nematocysts. So these are just three different kinds of nematocysts that you might find in an anthozoan like nematostella. And it just highlights there are differences in what the capsules look like. Um, and nematocysts might all be the same thing because they are shared uniquely among cnidarians. They're all made of certain kinds of mini collagens, but they're highly diverse in structure, in not just whether the tubule has spines or harpoons on it, but how it's put together, how it's packed in the capsule. And there's no overlap across the phylum in terms of specific capsule morphologies. There's not one kind of nematocyst that every cnidarian clade has. There's just this weird mosaic. So Leslie, doing CRISPR, made nematocyst mutants by accident. So what we found when we looked closely is that by turning off NV-SOX2, you can make a different kind of nematocyst in nematostella. You can create homeotic mutants that express a kind of nematocyst that nematostella will not make on its own, right? So what basically happens is the relative amount of tubule versus thread totally changes. You turn one kind of nematocyst into another. This is excellent evidence of homology, right? If you can turn these, just like homeotic mutants like Antennapedia, where you can see the limbs of fruit flies being ectopically expressed to turn into other kinds of limbs, you can do that apparently with nematocysts. The first and best evidence that the delivery structures, at least some of them, are homologous because you can make one become another if you manipulate the genes. Fascinating, really, really exciting, fascinating work. So this set of new tools that we have to study molecular biology and genetics, high throughput tools in things like proteomics, have really expanded the study of venom. So you can see in the 70s, there was a lot of study of venom of other cnidarians, which really just means jellyfish. And starting in the late 70s, as there were more molecular biology tools, people really began to study sea anemones. Not because sea anemone venom is like a pressing human health problem, but because sea anemone venom seems to be more biochemically stable. And so some of the technologies and tools that were available in the 70s, 80s, and 90s, you could extract that venom and put it on a benchtop and it wouldn't rapidly become something else or degrade. So it was possible to study it. So until about 2005, most studies of venom that were biochemical were on anemones. Once we got higher throughput, faster tools and better lab equipment, people went back to studying the venom of more dangerous animals. So I'm going to just orient you here with a phylogeny. This is a phylogeny of Cnidaria. Hexacoralia, which is the class to which sea anemones belong, is there in green. Anemones are there. They're about half the species diversity in Hexacoralia. So other hexacorallians, corals, black corals, and then things like button polyps, that's the other half. They are exclusively solitary. So coloniality is rampant among hexacorals, but not anemones. And anemones show the greatest habitat diversity among all anthozoans, in fact, not just among hexacorallians. We find them in the deep sea. We find them basically living on land because they can live in the high intertidal where they're exposed more often than they're wet. We find them in the polar seas, collections made at the North Pole and at the South Pole include anemones. They can live in fresher habitats than any other anthozoan. So they can live in about 1/4 seawater. So brackish environments, there are species of sea anemones there. They can also live in hypersaline environments. So they are really, really broad in their physiology. They use venom for everything. So they use venom to capture prey, usually using their tentacles. This marine sea slug has been captured and stuffed in the sea anemone's hole. That crab is stuffed in the hole and is being digested. So they also use venom on the inside of the body. Nematocysts and secretory cells on the inside are used in digestion because they don't have a stomach that they can pump digestive secretions into. They just use venom for everything. They also use venom in defense of themselves and in defense of their symbionts. So symbiosis is a huge and fascinating theme in cnidarian biology and especially in sea anemone biology. And it's the nematocysts that make the fish want to hang out with them. That crab there is called a boxer crab because they put anemones on their claws and then they fight with them. There's a shrimp, actually multiple shrimp in the tentacles of sea anemones. This is one of my favorite animals, Caliactus. The hermit crab has a sea anemone covering the gastropod shell that it lives in. And this animal has the coolest defensive interactions for their hermit crab. So the only thing that eats hermit crabs are cephalopods, really. You know, birds will pick at them, but cephalopods will take them, put them in their mouth, crunch them up with their beaks, and digest them. So that is the primary predator for hermit crabs. Cephalopods are visual predators and they're smart, so they learn. So what these animals do is if a cephalopod picks up a hermit crab plus anemone, the anemone releases brightly colored strings that are just basically 100% nematocysts. So those pink things poking through those what are white dots on the animal in the top picture are just packed with nematocysts. And it stings the bejesus out of the mouth of that cephalopod. The cephalopod spits it out, looks at what happened, presumably clocks that it's bright pink and that it all looks like this, and then doesn't do it again. They learn not to do this because it's not a good experience. So this is really important in defense, and there's fascinating behavior. There's probably some cool evolutionary stuff going on. I had studied these from museum specimens, which are brown, and the first time I saw them in the field, I had to do the very sad thing, which is to pull the hermit crab out of the anemone so that I could harvest the anemones. And when I pulled the hermit crab out, what I noticed was the hermit crab's abdomen has white spots and pink stripes. If you pull the hermit crab out, it looks like an anemone. So that if they are ever out without their shells, any cephalopod that has already learned will not eat them. So that is a really highly evolved, tightly interwoven system, just fascinating. The other amazing thing that sea anemones do with their venom that is very rare among animals is to use them in intraspecific interactions. So intraspecific competition in anemones happens slow, so it's not for any of you speed junkies. But what they use are specialized structures here in blue in these actinia, and they take these structures, differentially inflate them, and apply them onto another anemone. When they do that, little bits of tissue get pulled off of them. So they sacrifice tissue on those blue structures, basically suture it on to the red anemone there. And in suturing it on, they provoke an immune response in the victim. And that causes necrosis and cell death and ultimately animal death. So they use it in self, not self, aggressive interactions over space. And they have this has evolved twice within sea anemones. So in these structures called Ecology, which are, I always use myself like I'm a sea anemone as a diagram, but they are oriented completely differently. But they're at the distal end of the column, just under the tentacles. And then elsewhere, in a completely different lineage of sea anemones, they have evolved special tentacles called catch tentacles. They're thicker and bigger, and they use them the same way. They're like the finger of justice. They inflate these and then they apply them on someone they want to move. They suture a little bit of their tissue onto them, and then that thing slowly, like it takes a couple days, dies. And then when it dies, they take their space. So this is really amazing. It evolved twice in anemones. There's only one other case outside of cnidarians where this use of venom exists. It's in the platypus. So platypus use venom only in intraspecific competition. The males have a spur and a gland on their back leg, and they basically, I couldn't find a picture of it, but they basically do leg wrestling, which you can find a picture of on the internet. And they leg wrestle to impress the female platypuses. And so this is a known use of venom, super rare outside of cnidarians. Cnidarians, multiple times within a single lineage, evolved the ability to do this. So amazing, incredibly broad use of venom. So many of you might be thinking, okay, if their venom is so great, Meg, how come I can't feel it? Well, there's two reasons you can't feel it. Most of the anemones you touch don't make venom that is for catching or defending anything like a vertebrate, right? Mostly they're interacting with other invertebrates, the nervous system, and we see in many venomous lineages, vertebrate versus invertebrate venoms tend to be different. So mostly they're interested in microcrustaceans and worms and mollusks, and you are not that. So the venom is for a neuromuscular system that is not yours. Also, your fingers have a lot of callus. And so if you are touching the outside of an anemone with your finger, you're not going to feel very much. If, and people have, you touch the sea anemone to less callous parts of your body, even in things that you might encounter here, you will feel it, especially if you, the inside of your arm, especially if you were to test that against the digestive venoms inside the animal. So those tend to be, we think, those are broader acting and sort of more cytolytic and just punch through everything and you will feel it. So you can't feel the sting from an anemone because it doesn't want you to. It's not after you. Not all of them, but most of them. So when you study venom, there's a bunch of, these are just different ways. You can milk a snake, you can harpoon, you can milk a cone snail, you can milk a spider. Basically, you take the business end, the fang, the spine, and you use electrostimulation or dissection to remove the gland. It's not possible to do that in sea anemones. You can electrocute a whole sea anemone, basically just run a current through some water with a sea anemone in it and collect venomy mucus or venomy water. That's messy, and I'll talk a little bit more about how that might be problematic, but that is hard because they have this distributed system. It's super hard to just get venom from one part of the animal. You either have to get all of it, or you have to do what we do in my lab. This is my advertisement for my field program. This is my student, Alonso Delgado. This is us in Croatia. He's snorkeling, and then we put stuff in RNA later, and then we go to another beach and do this again and again. It's pretty hard work. It's actually pretty wonderful. So we typically study venom using transcriptomes because that's the most accessible way for us to get data, and because I'm interested in body part specific expression of venom, and you cannot get that very easily through proteomic methods. So we use transcriptomic methods. And there's really this, in venom in general, you can either look at sort of transcriptome, genome, or secreted venom, proteomes. What's super interesting in anemones is you don't get the same things when you look at secreted venoms versus transcriptomes. This is from some work done in collaboration with Pete Prentice's lab, and in particular with a grad student named Lauren Ashwood. So in yellow and in green are the things that they get that you can recover from a secreted study. In blue are the things you get from a transcribed study of venom. There is overlap, but it is not identical. There are things you get from secreted venom, and there are things you get from transcribed venom. One method does not get you everything. Presumably it's all there in the genome, but because some venoms are bound in nematocysts, they don't secrete very easily. When you electrocute that sea anemone, not all of the nematocysts fire. Some kinds of nematocysts probably don't fire. And so any venom associated with those, inside those capsules, you will not get. Likewise, transcriptomes are only going to capture actively transcribed genes. And so those threads of death that the sea anemone uses against cephalopods, those are not transcriptomically active. Studies, again, done with Lauren, showed that you get nothing out of the transcriptomes of Acontia, ever. Those nematocysts are made, the secretory cells are locked and loaded, they are not making more. If they make more, they make it elsewhere and then export it there. So they're not making new venoms in those structures that we know are full of venoms. So you can't get those in your transcriptome. So these two different, we go into this acknowledging we are looking at a partial picture. As I said, it's those, this is another animal and they're orange instead of hot pink, but we know we aren't getting, in the transcriptome, we are not getting those. We've tried really hard. And you can, but we know they're full of venom. So that's a close-up of one of the acanthia. The pictures on your far side of the room there, those are all the nematocysts, just a smash. It's just all nematocysts. It's like just nematocyst, nematocyst, nematocyst. You can see it also when you're looking at the section. It's all nematocysts, but you get nothing out of it. No venom if you do a transcriptome, which is crazy. We know it's there. So when you're understanding venom profiles, especially when you're using transcriptomes, but also when you're doing proteomes, you use functional assignment. It's an inference based on validated work done either in other taxa or by other studies. So we know that we think this is venomous because when we get a highly similar molecule out of a black widow spider, it kills mice, right? So we think this molecule that has the same chemical structure, that may be part of the same gene family, we think that it's a venom. So it's an inference based on similarity to a molecule found in a system that's validated. Homology, or inferences about similarity and evolutionary relationship, guides a lot of this. And that is tricky because highly modified molecules might not be similar anymore. So if there's been lots of evolution, you don't recognize that things are the same anymore. Or if you're trying to compare over lots of evolutionary time, which is the problem we have when we're studying cnidarians. So the age of the lineage Cnidaria is equal in age to its sister. So it is as old as all triploblasts, about 800 million years old. Anemones themselves have been dated to about 650 million years. That's very old. Snakes as a clade are less than 100 million years old. Cone snails are about 100 million years old. This is a very old system. It would be like comparing the venom of fish to the venom of slow lorises, right? That is about the evolutionary divergence. We're not expecting identity across those systems, given all the evolutionary divergence. So that is always in our mind as we're doing this work. And we also know you can have molecules that are chemically similar, that seem to have similar, would presumably act in a similar way in a biochemical pathway, and they could come from completely different gene families. So genes in the same gene family might not be venomous. Things that have the same kind of biochemical profile might not be related. So we go into this knowing that. This is a series of venom profiles. So the taxa underneath the blue bar are part of the lineage we call Actinioidea. These are most of the kind of familiar tropical shallow water anemones. Those under the purple bar is a lineage called Metridioidea, deeper water, colder water, a few things. Most of the invasive species of anemones are in that lineage. So two different lineages. The green one is the model system Nematostella. It's part of the third major lineage of anemones. And within each of these taxa, what you're seeing is the venom phenotype, broken down by functional category of the transcribed venom. So you can see that everybody has all of these different categories of venom. They're making some kind of allergen and innate immunity, making some things that are acting as cytolytic toxins, hemostatic toxins. Auxiliary, these are things like PLA2s that in other taxa act in conjunction with one of these other kinds of venom. So these are what venom phenotype profiles look like. And you can generate these from transcriptomic data by searching for genes that have been annotated to have venom function. We know we're not catching everything because it presupposes that you're on a fishing expedition using known things as your guide. But that's what we have. So the questions, given all this background, I'm going to talk about two quick questions. One is looking at how coevolution impacts venom. So this is work, again, my PhD student Alonso Delgado has been doing. We did it in collaboration with my former student, Jason McCrander. And we were using some existing data on clownfish hosting sea anemones, which, as you all know from the work happening in Ben's lab, has evolved multiple times. So we see this association evolving multiple times independently. So a question you might want to ask as you're looking at these taxa is, does living in association with clownfish impact venom? Every time this has evolved, are there similar signatures in the venom? So taking existing transcriptomic data, and these are all interestingly about the same age, which is always nice when you're making these evolutionary comparisons because you're more like comparing apples to apples. If we look at the venom profiles, we can see of uh, exemplars from these lineages, we can see that there is variation in them, right? So here you can see all the different things that they do. They're all doing all the kinds of things, but there are differences in the relative investment. And so here I have them bunched up based on their lineage. And they don't seem, there is not kind of a super strong signature as you look at them, you know, it doesn't seem like within the clades, they are super strong in terms of their affinity or similarity. When we compare them across other anemones and ask, are they more similar? They evolved independently. Are they more similar than we expect, given the diversity seen outside of these groups? What we find is that there are more hemotoxins, more blood system dysregulators, and less neurotoxins. So that's interesting. We don't really know what it means, but we see this pattern. More hemotoxins, less neurotoxins. So this may be part of their strategy for making better defense. It may be part of these may be molecules that are easier to regulate the discharge of, and so this may be a safer way to associate with fish. We don't really know what it means, but we do see this consistent pattern of more hemotoxins, less neurotoxins. So another question we have asked is about spatial differentiation. This is Alicia, which has these, they're sometimes called berries, these clusters, brightly colored on their body. These are packed full of nematocysts. We think they are defensive or something. We don't know what they do, but these are zippy. If you touch them, you will feel it, even with your hand. But you are likely to encounter these diving. They live at diving depths. And if you get close to them, they kick off a lot of mucus. That mucus hits your face. You know you have found them. So we are curious about these clearly visible regions in this animal and in others and what that looks like in terms of their venom. So work that Lauren did as part of her PhD was to take a single species and in some cases a single animal and to look at it through a variety of perspectives. So that is a diagram of the animal. This histological section is next to it. What you see underneath is a kind of heat map of gene expression. All of those acanthia are not hot. They are not one of the bright colors. And when you look at the different regions of the body, tentacles, which we think are involved in defense and predation, gastrodermis, that's the inside of the animal, we think it's involved in digestion, and epidermis, which should be defense on the outside of the body, you can see the transcriptomes look different. The areas, the part of those heat maps that are lit up differs and is differentiatable across body regions. So it's not just specialized structures, but whole regions of the body are transcriptomically different. Now, whether those elements are venoms or not is something that is not resolved at this level yet. But where we have resolved it is in studying these self, not self, intraspecific aggressive structures called Ecology. So here you can see, listen, I have an older sister and we spent many car trips in the back kind of slapping each other. That's what these two anemones are doing with these inflatable structures. And then they, as I said, they create these pockets of cell death. So for Jason's PhD, we could dissect off those akaraji. They're big. And so we would stimulate the animals so that they would make a lot of akaraji. They'd be ready to fight. They'd be growing the akaraji because we were kind of putting the two anemones next to each other. And so we knew they were transcriptomically active in the akaraji. And then when they'd start to fight, we'd cut them off and flash freeze them and then look at transcriptomes. They regenerate. It was okay. They lived in the lab for years. So that's such a plus of working with cnidarians. You can do this kind of Prometheus thing where you pick at them and they still live. So we did that with these animals. It's great for replicates because you can let them heal and then do this again. But what we found was that we had Akaraji expression of venom genes, including some newly identified molecules. And these are cytolysins. Cell death made a lot of sense. What was fascinating for us when we did this tree, the things that we found are there in bold. The genes most similar to it were, whoops, were from not a close relative, but another animal that uses this self, not self, interspecific aggression. So Sagartia rosea makes catch tentacles. We presume that these are genes expressed in catch tentacles because otherwise Anthoplura is much more closely related to these other actiniids than it is to Sagartia. They are not closely related at all, like 500 million years of separation. So about as closely related to them as you are to a frog. So among all these other things that are less than 100 million years of evolutionary relatedness, these two things group together. We think that this is a molecule associated with self, not self, venom. This kind of use of venom as I said, it's unusual. It's super interesting that it's an example of this deep homology. They are clearly activating common pathways of genes in different structures, just like opsins get expressed in different places to make eyes. So it's a kind of example, very much like what we see in opsins. So it's not Akaraji expression of genes, it's interspecific associated expression of genes. So venom evolves rapidly and under dynamic selection. This is the dogma, kind of central dogma of venom evolution, right? It's this rapid response trait, and there's this co-evolution of predator and prey, and some beautiful examples of snakes co-evolving with mammal prey and mammals evolving usually through von Willebrand factor associated genes, ways of detoxifying venom so that they have a chance at survival. It's this very dynamic system. We also see something very similar in cone snails, particularly in cone snails and their interactions with worms and with fish. So that's the paradigm for venom evolution. Rapid, highly targeted, lots of positive selection, dynamic system. But this is in cases where venom is very targeted and relatively young. So we've been looking at what might be happening in systems where venom is used for everything, right? It is the Windex of the biochemical array, right? For cnidarians, they use it for everything. They're not just using it to capture their food. They are using it for prey, for digestion, for aggression, for defense, everything. So here again is functional groups looking at a bigger array of anemones. This is work that we have out for review right now. So taking those functional profiles very broadly across anemones. And I think the take home, when you really look at this, is at the level of the functions that venom is expected to perform, everybody does everything. There is high conservation of functional breadth across anemones. You have to be able to use it for digestion because that's all you've got. So unlike venom in snakes, there isn't specialization at the level of predatory, or in cone snails, predatory venoms. You have to be able to do predation, digestion, all of these functions with venom. And so that means you need to conserve all of these different categories of function. But when we look more closely, so this sample, this is, as I said, work that's more recent. We have a broader and evolutionarily more representative sample. So we have this remarkable stability. We see variation in relative expression. So when you look not at does it make a cytolysin, yes or no, but like, is it amping out on cytolysins or is it amping out on neurotoxins? There are differences. And those differences are interestingly spread across the tree, right? There does not seem to be lineage-specific specialization in functional categories. Other work that we've looked into and that colleagues have looked into have found that across lineages, they might be making more neurotoxins by increasing copy number, for example, of one kind of neurotoxin. Or so we'll see rapid evolution in one element within these functional profiles differently across the whole clade. So much more dynamic than what is happening canonically in terms of all the opportunities to specialize and customize while still conserving broad functional capability. So getting back to this question I asked, I started asking about 15 years ago, is venom different in anemones? The short answer is yes, it's really different. And I would say, really, venom is different in snakes than it is in cone snails, than it probably is in all of those other lineages of the tree. Anemone may not be an outlier if we knew everything about everything. But what we see is that venom phenotype is much less variable. At the phenotype level of what is the venom capable of doing, it is broad scale and there's high conservation. Dosage and kind of within those functions, dosage and approach is pretty variable. And copy number is probably a factor here. So making more copies and having different isoforms of the same genes might be a way. They can't store venom. So they have to have a way of rapidly expressing, rapidly refilling all those secretory cells. One way of doing that is through copy number. So there's some genomic things that are probably driving or contributing to this system. When we look at the actual genes, which I didn't talk about here, we compare copies and we calibrate rates. The anemone scale comparisons find low negative selection, right? They don't find the positive selection we see in snakes. We see conservation rather than variation. When we look within species, though, we see lots of variation. So that disconnect between the variation we see within clades and within individuals and what we see across the group is because when you're looking across 700 million years of evolution, the only things you can find to compare across those endpoints are things that are highly conserved, right? Just like when we compare humans and mice and chickens and marine sponges, we use genes like 18S that are highly conserved. We know that they're highly conserved. That's why they are findable and comparable across that breadth of time. So it's the same principle when we're comparing neurotoxins across anemones. Only ones we can compare across that swath of time are the genes that are highly conserved. If they weren't, they'd be too variable and we couldn't use homology methods. So it's a tautology that these broadly shared genes are highly conserved. It's just necessary by our methods. But what this suggests is that there's a lot more variation to discover and a lot more complexity. So why does this matter? Well, turns out anemone venoms are pretty badass. They do really cool things. There are some clinical trials on using sea anemone venoms to address autoimmune disorders. There are sea anemone venoms that are really potent, directed cell killers. So there are therapeutics that benefit from understanding the breadth of function and diversity of sea anemone molecules. But it also suggests some cool things when we think about anemone biology. So anemones have this incredible success and longevity, right? All marine habitats, all depths. They're symbiotic with a whole array of things from poriferans, crustaceans, mollusks, other cnidarians, ctenophores. You think that's a ctenophore, and it is, but there's a sea anemone parasitic inside of it. Fish, algae, dinoflagellates, a huge array of interactions. They have diet breadth from vertebrates to dissolved organic material. They have a lifespan of months to centuries. They have this incredible lineage age. Venom is a part of all of this. The venoms that are highly conserved and functionally constrained probably contribute to some of these factors, and the venoms that are more dynamic probably contribute to others. So it makes sense that this picture, these extraordinary factors have to do with venom, because venom is at the heart of how anemones interact with each other and with the world. That's the sea anemone that lives in ice. We don't really understand how it does that. If you put a sea anemone in ice in your freezer, that's freshwater ice also. There's so much about that we don't understand. It's hanging upside down, living in ice. we don't know what's going on there, but they do all this stuff. We still don't understand so much of it, which is so exciting. It just suggests that the world, so many things that we've known about for hundreds of years, still have so much to teach us. And the world is just much more interesting and complicated than it would seem to be. So this work is highly collaborative with, that's Ben there when he was a graduate student, with lots of colleagues and with lots of support from the National Science Foundation. And I will stop there.